When assessing the health and well-being of a pregnancy, several different medical tests can be used to look for birth defects and assess the overall health of the fetus. Fetal ultrasonography is often used along with these prenatal diagnostic tests to give us better, more accurate information. There are 3 tests that we're going to discuss in this presentation. The first is amniocentesis. Amniocentesis is a process that removes a small amount of amniotic fluid from the amniotic sac. To avoid rupturing the amniotic sac, the needle is often administered through the abdominal wall. And you can see amniocentesis shown right here, with the needle going through the abdominal wall, through the uterus, and into the amniotic cavity. Ultrasound is going to be used to ensure that the needle doesn't damage the fetus or the umbilical cord. But in the event that somebody doesn't want to use this procedure, we also have another procedure known as chorionic villi sampling. Chorionic villi sampling can actually be administered a little earlier than amniocentesis, and it does tend to be a little less invasive. This procedure includes taking this catheter, inserting it in the vagina through the cervix and up to the uterus to collect a sample of tissue from the chorionic villi. This test will give you the exact same information that amniocentesis will give you as well. The other test that we're going to discuss is the maternal alpha-fetoprotein test. AFP is produced by the fetus and released into the mother's blood throughout the pregnancy. Levels of AFP can be assessed by taking a blood sample from the mother. And while we can't get as much information from this procedure as we do from amniocentesis and chorionic villi sampling, we can still use this test as a marker for different birth defects. AFP is going to be released into the mother's bloodstream till about 15 weeks. After the 16th week, there shouldn't be any more AFP levels inside of the blood. So we can use these levels to determine the overall health of the fetus. Higher levels of AFP after this 15th week can indicate different types of birth defects. Now, as we're all aware, as pregnancy progresses, there are going to be a lot of changes that are occurring in the fetus. But that being said, the mother is also going to undergo a fair share of changes as well. One of the most apparent changes is the development of the placenta. We already know that during the first 3 to 4 months, the corpus luteum is going to be releasing estrogen and progesterone. This continues the growth and maintains the endometrium. And at the same time, the chorion, which is going to be derived from the trophoblast, is going to release another hormone known as human chorionic gonadotropin, or HCG. This is going to feed back on the corpus luteum to continue its release of estrogen and progesterone. This ensures that menstruation does not take place, and the blastocyst that's implanted into the endometrium stays attached to the mother. But after about the 3rd month, the placenta is going to start releasing hormones inside of the mother as well. You can see a synopsis of all these hormones that are being released by the placenta shown right here. Human chorionic gonadotropin, or HCG, is going to feed back on the corpus luteum and cause it to continue its release of progesterone and estrogen. But we also must keep in mind that the corpus luteum also releases relaxin and inhibin as well. We know that progesterone is going to maintain the endometrium, and both of these hormones are also going to help prepare for lactation as well as prepare the mother's body for the birth. Relaxin is also going to help prepare the mother for birth, as this is going to increase the flexibility of the pubic symphysis and also relax uterine smooth muscles and help to dilate the cervix. But there are 2 other hormones that are coming here from the placenta, including human chorionic somatomamotropin and corticotropin releasing hormone. I'm going to discuss these 2 hormones here in just a few slides, but I just wanted to quickly mention that human chorionic somatomamotropin, or HCS, is going to work with estrogen and progesterone to prepare the mammary glands for lactation, and this is also going to amp up the metabolism by increasing protein synthesis and also increasing ATP production from fatty acids. HCS will be coming from the placenta just as the rest of these. Now we also have corticotropin releasing hormone coming from the placenta as well. And this is a bit odd because we've seen corticotropin releasing hormone before coming from the hypothalamus. So what on earth is it doing here coming from the placenta? Well, this is in fact going to be released by the placenta, and this is going to do a couple things for us. We know that corticotropin releasing hormone from the hypothalamus is going to make its way down the hypophyseal portal system into the anterior pituitary gland to affect corticotrophs to release adrenocorticotropic hormone. ACTH is going to go down to the adrenal cortex and lead to the release of cortisol. So corticotropin-releasing hormone from the placenta is in fact going to do the exact same thing. Cortisol is the stress hormone, and labor is a very stressful process, so we want our cortisol levels to be a little higher. But corticotropin-releasing hormone also establishes the timing of birth. This acts as a clock that tells the body when it's time to go into labor. You can see here in early pregnancy the HCG levels are going to spike, and then here by the 3rd month the placenta is also going to start releasing estrogen and progesterone. So we see those levels increasing drastically as this process continues. This slide will just review the actions of relaxin and HCS. Relaxin, as we know, is released by the corpus luteum, and it's going to increase pubic symphysis flexibility and also loosen up some of the joints around the pelvic region as well, to loosen up those bones to help the baby squeeze out much easier. Human chorionic somatomammotropin is going to help prepare the mammary glands for lactation, and it's also going to be a metabolic regulator in both the mother and in the fetus. Corticotropin-releasing hormone is going to release cortisol. But one other action that CRH is thought to have is that this is actually going to help develop the fetal lungs and also produce surfactant. Surfactant is a fluid that's released by the lungs that's going to cut down on surface tension. We must remember that surface tension is going to keep the lungs from opening up, and without surfactant, breathing is very, very difficult. So this is going to help produce surfactant, which will help the fetus be able to breathe whenever it leaves the mother's womb. Perhaps the most apparent maternal change that's going to take place during pregnancy is simply the enlargement of the uterus as a baby is going to be growing inside of it. The increase in contents in the abdominal cavity increases the pressure in the abdominal cavity as well. This is going to result in the digestive organs being pushed out of the way and extra pressure being put on the digestive organs as well. In the event that the stomach is being pushed up against, this can actually lead to gastroesophageal reflux, which is what causes heartburn. You can see on this picture an example of a very enlarged uterus and the digestive organs that have been moved out of the way. Now, there are many different changes, like I mentioned, that are taking place in the mother during pregnancy, and they don't stop at the ones that we've just discussed. Mothers will also experience morning sickness as well as postural changes due to the change in weight distribution because of the baby that's growing in the uterus. These factors also play into a mother's ability to exercise during pregnancy. Morning sickness is going to be a very potent deterrent to exercise, and with the postural changes and weight increases that pregnancy brings about, it can be very difficult to exercise as one did before pregnancy. One of the other things that's very relevant for us to mention here is that as relaxin levels go up, that's also going to loosen our joints and make the mother much less stable. So heavy lifting is often not recommended for pregnant women. Now, at the end of pregnancy, it will be time to get the baby out of the mother's womb, and that is the process we call labor. Labor is the actual expulsion of the fetus through the vagina. And there's actually a very interesting positive feedback loop that's going to regulate this process. I don't have that positive feedback loop posted in these slides, but during Anatomy and Physiology 1, when you would have discussed positive feedback loop mechanisms, this was the example that your textbook used. This example includes oxytocin being released, leading to smooth muscle contractions. That's going to cause the uterus to constrict, pushing the baby outwards, and the more constriction there is, the more stimulation of stretch receptors that we have. As contraction continues, the baby is going to be pushed out of the mother's womb, and once the baby is born, that interrupts the cycle, and therefore the positive feedback loop mechanism is going to cease. You can see here that labor is separated into 3 different stages: the stage of dilation, which we can see here, the stage of expulsion, which we can see here, and the placental stage, which we can see here. Now, the stage of dilation is when the cervix is going to dilate and the amniotic sac is going to break. This is going to cause amniotic fluid to leak out of the vagina, and this is going to indicate that it's time for the baby to be born. The stage of expulsion, which is the second stage here, is the actual pushing of the baby out of the vaginal cavity, but that is not going to be where labor ends. The placental stage is also going to need to take place, where the placenta will be shed from the uterus and then removed from the vagina as well. Now, one thing I do just want to mention about this picture is you can really see how much larger the uterus has become through the pregnancy. This is going to be something that's not going to go back to normal right away. Thinking about the length of pregnancy, the body has had 9 months to adapt to the changes of the growing fetus inside of it. Compared to the length of the pregnancy, labor is going to be a very, very fast process. So the uterus's reduction in size is referred to as involution, and that process is going to take about 6 weeks. Dystocia is a term to indicate difficulty in labor, and if severe enough, a cesarean section is often used to get the baby out of the mother. A cesarean section, or C-section, is when an incision is made in the abdominal wall of the mother, and the baby is then removed by that means instead of through the vagina. Now, the mother is not the only one that's going to be undergoing changes at the time of birth. The infant itself is going to start undergoing some various changes, as it's now going to be required to survive independent of the mother. It's extremely important that the baby's respiratory system and nervous system are properly functioning in order for it to survive. Without the ability to ventilate on its own, the baby is not going to be able to bring in adequate oxygen to perfuse its tissues. By about the end of the second trimester, or the 6th month, the lungs are at this point just about developed, and it is at this point that premature babies are generally able to survive on their own. The respiratory rate is generally going to decrease a couple weeks after birth, dropping from 45 breaths a minute to 12 breaths a minute after the 2nd week. Many of the apparent changes are in fact the changes that we discussed when we covered the fetal circulation. The foramen ovale is going to become the fossa ovalis, the ductus arteriosus is going to become the ligamentum arteriosum, we have the ductus venosus, which is going to become the ligamentum venosum, and then the umbilical arteries and the umbilical veins are going to become the medial umbilical ligaments and the ligamentum teres, respectively. While these structures were providing a very important function when the baby was in the mother's uterus, once the baby is born, it's very important that the baby is able to have a proper cardiovascular system and that these structures are going to close up. Now, after birth, thanks to the hormonal influences in the mother, lactation is going to take place, which refers to both the production and ejection of milk from the mammary glands. We have 2 different hormones that are going to control lactation, namely prolactin and oxytocin. Oxytocin was helping to constrict uterine smooth muscle during labor. Here, it's going to constrict myoepithelial cells that are going to lead to the ejection of milk from the nipples. Prolactin is the hormone that's going to be responsible for producing milk, and oxytocin is going to be responsible for ejecting it. Prolactin came from the anterior pituitary, and oxytocin came from the posterior pituitary. Let's take a look at this feedback loop, including oxytocin leading to the ejection of milk from the nipples. So first and foremost, the baby is going to start suckling on the nipple. That's going to increase touch sensations in the nipples and activate sensitive neurons in the nipples as well. Those are going to send signals up to the hypothalamus, which is going to increase the production and release of oxytocin. Oxytocin is going to lead to the constriction of myoepithelial cells in the mammary glands, and that's going to lead to the milk ejection. Now, this is a positive feedback loop mechanism. So, the more milk that's present, the more suckling that takes place, and the more stimulation there's going to be. That's going to lead to more oxytocin release until the baby stops suckling on the nipple. There seems to be a lot of debate these days going around about the efficacy of breastfeeding versus bottle feeding. But your textbook will tell you that breastfeeding is definitely the superior option. The reason for this is because the mother's milk has all of the vital nutrients and chemicals that the baby's going to need for development. We will also see white blood cells found in the milk, and also antibodies that are transferred to the fetus through breastfeeding as well. This is going to be very important for our natural passive immunity. The mother's body produces the breast milk to ensure the optimal growth of the infant. Now, the last topic that we're going to cover in this chapter is inheritance. Inheritance refers to the ways that traits are passed on from parents to offspring. And genetics is the study of how inheritance takes place. For starters, with inheritance, we must keep in mind that humans have 23 pairs of homologous chromosomes. This means that 23 chromosomes come from mom, 23 chromosomes come from dad, and mom and dad's chromosomes will be homologous to each other. Mom's chromosome 1 is going to contain all of the genes that dad's chromosome 1 is going to have, but the versions of those genes are what are actually going to be different. A chromosome is essentially a bunched up, very long strand of DNA, and genes are segments of those chromosomes. All genes for the same traits are found in the same locations on each homologous chromosome in both mom and dad's set of of chromosomes. Now, the reason that we can pass on different traits to our offspring is that there are different versions of every gene in the human. Those different versions are referred to as alleles. So just for the sake of an example, there might be the allele for brown hair, and there will also be the allele for blonde hair. A mutation is any permanent change in any allele, and oftentimes we tend to think of mutations as a bad thing, but this is not actually always the case. Mutations can be non-beneficial, and they can even be detrimental, in the event that the mutation took place in an allele that was coding for an enzyme that our body desperately needed. But oftentimes, mutations are changes in alleles that actually benefit a particular member of the species and give that member an evolutionary advantage. A few other terms before we jump into an example of a mutation are genotype and phenotype. The genotype refers to the combination of alleles that one member of a particular species has. Oftentimes, either mom or dad's allele will be what's known as the dominant allele, which means that that allele is going to mask the presence of the other allele, and the other allele, which is the recessive allele, will not be expressed in the offspring. Generally, we depict the dominant allele with an uppercase letter and the recessive allele with a lowercase letter. So, if mom has passed on a dominant allele and dad has passed on a recessive allele, we would put one uppercase letter next to a lowercase letter to depict what genotype we have. Genotype is the actual genetic makeup that pertains to a particular trait that we're talking about. Now, the phenotype, on the other hand, is the actual physical expression of whatever the allele is. So, if mom passes on the brown hair allele and dad passes on the blonde hair allele, but the brown-haired allele is dominant to the blonde-haired allele, the physical expression would be the phenotype. The genotype would state that we have both one allele from mom and from dad, and one of those alleles codes for brown hair, and the other allele codes for blonde hair. But the phenotype is the physical expression, so that's going to be simply what that particular offspring is displaying. So the example that we're going to look at here to demonstrate this idea is with phenylketonuria. Phenylketonuria is a condition where the enzyme phenylalanine hydroxylase cannot be synthesized. Phenylalanine hydroxylase breaks down the amino acid phenylalanine. So individuals with phenylketonuria have to be very careful about eating phenylalanine. We're going to use a Punnett square to walk ourselves through this example. So what we're seeing here is a set of homologous chromosomes from the father and homologous chromosomes in the mother. We want to keep in mind that the father is going to have his own homologous chromosomes, and the mother is going to have her own homologous chromosomes. So you can see that this allele is the dominant allele, and this one would be recessive. This one would be dominant, and this one would be recessive as well. Now these two carriers happen to be heterozygous, which means that they contain one dominant allele and one recessive allele. It's also possible for one carrier to contain two dominant alleles and two recessive alleles. Those individuals we would call homozygous dominant if those two alleles are dominant, and homozygous recessive if those alleles are recessive. In the event that two heterozygous mates come together, we have the possibility of several offspring. We could get this egg cell that contains a dominant allele and this sperm cell that contains a dominant allele, and that would give us a homozygous dominant offspring. We can also have this sperm cell with a dominant allele fertilizing this egg cell with a recessive allele, and that's going to give us a heterozygous offspring. And we're going to get the same result in the event that this sperm cell was to fertilize this egg cell. So both of these would be heterozygous offspring. This individual would be formed when this sperm cell meets up with this egg cell. So what we're seeing here is the possible genotypes of all of the offspring of these two individuals. Now we've seen the genotypes. We've got homozygous dominant, heterozygous dominant, and homozygous recessive. If these two individuals continue to mate, 25% of their offspring are going to be homozygous dominant, 50% are going to be heterozygous dominant, and 20% are going to be homozygous recessive. But we want to keep in mind that the dominant allele is going to overpower the recessive allele. And the example that we're utilizing here is phenylketonuria. The dominant allele indicates that the offspring will not have phenylketonuria. But this recessive allele is going to code for a defective phenylalanine hydroxylase enzyme and lead the individual to have phenylketonuria. So in terms of the genotype, as long as we have one dominant allele, we're not going to have phenylketonuria. So these individuals, the homozygous dominant and the heterozygous dominant, will not have phenylketonuria. But in the event that we have a homozygous recessive carrier that ends up with 2 recessive alleles, this individual will have phenylketonuria. A few other examples of traits that are passed on with this simple dominant recessive inheritance pattern is albinism, myopia, hyperopia, diabetes insipidus, Huntington's disease, and a widow's peak. These traits are quite diverse, but they're all passed on in the ways that were just described. Now, aside from the traits that I just showed you, in fact, most of our traits are not inherited in this simple dominant recessive inheritance pattern. There's often many other factors that are going to play in to which traits are expressed in the offspring. Incomplete dominance refers to a situation where neither allele is dominant over the other. Essentially, the mother's allele is going to be expressed and the father's allele is going to be expressed. And an example of this is with sickle cell anemia. Sickle cell anemia is caused by a defective type of hemoglobin, specifically hemoglobin S. So eventually, the father's homologous chromosomes will be separated into 2 different sperm cells, and the mother's homologous chromosomes will be separated into 2 different egg cells. Hemoglobin A codes for the normal hemoglobin, and hemoglobin S is going to code for the sickle cell hemoglobin that's defective. In the event that we have 2 heterozygous carriers, we're going to have the same distribution as we saw with phenylketonuria. The difference is, because we're talking about incomplete dominance, these phenotypes are going to be a little bit different. You can see that the normal genotype with 2 hemoglobin A's is going to leave the offspring normal. And the homozygous sickle cell hemoglobin is going to leave the patient with sickle cell disease. This is going to result in severe anemia and many other health issues. But in the event that we get a heterozygous carrier that has 1 copy of the hemoglobin A gene and 1 copy of the hemoglobin S gene, this individual is just going to be a carrier. In this person's bloodstream, they're going to have normal hemoglobin and also sickle cell hemoglobin. And while this does leave the offspring with some of the effects of sickle cell, this also confers a resistance to malaria. Another inheritance pattern that we're going to see is multiple allele inheritance, which is when the gene has more than just 2 forms. We're going to use the blood groups to give us an example of codominance. With codominance, both the mother's alleles and the father's alleles are going to be expressed. Essentially, there's 6 different genotypes that can be found. 1 allele is going to code for the A antigen, 1 allele is going to code for the B antigen, and 1 allele is going to code for no antigens at all. And based on the mixture of those alleles from both mom and dad, that's where we get our different blood types. I do encourage you to go into your textbook and read about this example, as there's going to be much more information there than I have on this slide. But this is another type of inheritance pattern that we will see, known as codominance. Now truly, many different traits are passed on through polygenic inheritance, which is when the presence of a trait is controlled by more than 1 gene. We can also take this a step further and state that complex inheritance is where traits are controlled by more than 1 gene, but also by environmental factors. And these are all examples of complex traits that are passed on from complex inheritance. Skin color is a great example of this. There are several different genes that are going to code for skin color. But on top of this, the amount of time we spend in the sun is also going to affect our skin color as well. Height is also another factor that's passed on through complex inheritance, as there's many different genes that code for height. But in the event that we don't get all of the nutrients that we need when we're young, we're not going to grow to our full height potential. This takes into account environmental factors as well. Hair color, eye color, metabolic rate, and our overall build are all examples of traits that are passed on through complex inheritance. Now, we should be experts on the fact that humans have 23 pairs of chromosomes. There's a total of 46 chromosomes, as both mom and dad's chromosomes make 23 pairs. A karyotype is when we take both sets of these chromosomes and arrange them in their proper order. That gives us the ability to see each homologous chromosome, and also shows us that the 23rd chromosome is quite unique. Chromosomes 1 through 22 are referred to as autosomes, because everybody's going to have the same autosomes. That being said, the 23rd set of chromosomes are the sex chromosomes, and their inheritance is going to determine what sex we are. The 23rd pair of chromosomes can either be X or Y. Males will have both an X and a Y chromosome, and females will have two X chromosomes. As I mentioned on the last slide, there are 22 autosomes and a single pair of sex chromosomes. Generally in females, one of the X chromosomes is going to be inactivated. And since males are the only ones that carry the Y chromosome, it's the sperm cell that's going to determine what the sex of the baby is. You can see here that we're going to have one sperm cell with an X chromosome and one sperm cell with a Y chromosome. And over here we're going to have two different eggs that both have the X chromosome that came from the mother's double X. Here you can see we have XX and XY. And this Y sperm cell has equal likelihood of fertilizing either this egg or this egg. So we end up with about half of the offspring being males and half of the offspring being females. Certain traits can also be sex-linked, which means that they're going to be found on the X chromosome. Sex-linked inheritance can be a little bit complicated, so let's go through this example and understand how this works. An example of a trait that's passed on that's sex-linked is red-green color blindness. Red-green color blindness is more often seen in males than in females. And it is because this trait is sex-linked that's the reason for this. This example gives us a normal male and a female who's carrying the recessive gene for red-green color blindness. This recessive gene is depicted as X with a lowercase c, and the normal allele is going to be depicted as X with an uppercase C. We see the normal allele shown over here in this normal male as well. When this sperm cell fertilizes either of these eggs, that's going to leave the offspring with at least one copy of this normal X chromosome. So neither of these individuals are going to have red-green color blindness. This individual will be a carrier, but it's not going to be expressed in the phenotype. Now, in the event that this sperm cell carrying the Y chromosome is going to fertilize either of these egg cells, here, this is going to leave us with a normal male. But if this sperm cell fertilizes this egg cell with the recessive red-green color blindness chromosome, this is going to result in a colorblind male. You can see that this offspring is a normal female with both sets of normal X chromosomes. This individual is a normal male, and we see that represented right here. So this individual that we see here, this normal female is not going to express red-green color blindness, but she is going to be a carrier for that trait and can possibly pass that trait on to her offspring. This individual over here is only going to have one copy of the X chromosome, and therefore we're going to get a colorblind male. It is for this reason that certain traits are more often seen in males compared to females. It is not impossible to get a colorblind female, however, we would have to have a red-green colorblind male mating with a female who's a carrier for the recessive trait. A few disorders to finish us off are infertility as well as some congenital defects. Infertility in males and females was briefly discussed in the last chapter. This is an overall inability to reproduce. Fertility does decrease with age, and this happens differently in males and in females. Females will have a limited number of egg cells, so into the later years, reproduction will not be possible. Infertility in males can be seen through erectile dysfunction, as well as due to low sperm counts. Now this term here, congenital defects, is a blanket term for any type of issue that is passed on in the chromosomes. This is any type of genetic defect that individuals are born with. Down syndrome is an example of a congenital defect, and the more accurate way to scientifically discuss Down syndrome is with its name, trisomy 21. Trisomy 21 indicates that there's 3 different copies of chromosome 21. This disorder is going to stem from a difficulty that occurs during meiosis. Mental retardation as well as stunted growth are both symptoms of Down syndrome. So that will conclude our final chapter here in Anatomy and Physiology 2. I want to congratulate you all on your hard work this semester and commend you all for making it to the end of the course. I know you've all worked extremely diligently this semester, and your hard work does not go unnoticed. If you have any questions regarding Chapter 29, I encourage you to reach out to me for assistance, and if you ever need to get in contact with me, please feel free to keep my email address. I wish you all well as you continue on in your education and your future careers, and if I can ever be of any assistance to you, please don't hesitate to reach out. I hope that this was an enjoyable semester for you all, and I definitely hope that you can say that you learned something this semester. I wish you all good fortune, and I wish you all the best going forwards. Thank you for your attention this semester.